My name is Noah Dickinson, and today I'll be discussing an ongoing project here at Nossum University, where we're trying to determine how the prostaglandin signaling system is involved in the development of radiation-resistant forms of breast cancer. I'd like to disclose that I'm a student at Laurentian University, working in collaboration with Nossum University, and that this study has received funding from an NSERC Discovery Grant, the Northern Cancer Foundation, the Nossum University Faculty Association Research Development Award, and the Canada Graduate Scholarship for a Master's Program. Our work uses MDA-MB231 cells as a model. These cells are a particularly aggressive form of triple-negative breast cancer and can become radiation resistant with recurring doses of radiation. In fact, our lab has previously done this and created radiation-resistant forms of these MDA-MB231 cells. During this previous work, a bulk sequencing approach and a whole transcriptome profiling approach were used to investigate new targets for treating and preventing these radiation-resistant variants. The results suggested that the prostaglandin system was heavily dysregulated. For numerous reasons, we initially predicted that prostaglandin endoperoxide synthase 2, also known as PTGS2, plays a particularly important role in the development of radiation resistance in breast cancer. It's the rate-limiting step within the prostaglandin signaling pathway, and it is also involved in the creation of 5 different prostaglandins, 3 of which have been demonstrated to have a significant impact on tumorigenesis, proliferation, angiogenesis, and metastasis, which all contribute to increased radiation resistance. More recently, we've been investigating this dysregulation in PTGS2. Using RNA sequencing, RT-qPCR, and Western blotting, we have observed a significant upregulation in PTGS2 in our radiation-resistant cells. We then used a CRISPR-Cas9 activation system to overexpress PTGS2 in our MDA-MB231 cells. This overexpression was confirmed by RT-qPCR and Western blotting. In each figure in this poster, AC denotes control cells with typical expression of PTGS2, and A1 and A2 denote the PTGS2 overexpressing cells. A clonogenic survival assay, post-radiation, was used to determine radiosensitivity, and we were really surprised by the results. Contrary to our initial prediction, it seems PTGS2 overexpression caused a significant decrease in survival post-irradiation, despite being overexpressed in our radiation-resistant variants. This likely indicates that either PTGS2 was accomplishing some other role aside from radiation resistance in the radio-resistant cells, or that some additional factors such as a change in expression of the downstream prostaglandins or the receptors may be required to affect the radiation resistance. Next, cell proliferation was assessed with a growth assay, in which no significant differences were observed between the PTGS2 overexpressing cells and our typical MDA-MB231 control. We also used RT-qPCR to examine 5 other genes associated with the epithelial to mesenchymal transition, which is a change in cell physiology associated with increased risk of metastasis. We found that fibronectin was being expressed roughly 3 times as much in the PTGS2 activated cells relative to the control. Additionally, when completing an adhesion assay, we found that the PTGS2 overexpressing cells were not binding as much to fibronectin as the control cells were, although more replicates of this assay are needed to ensure reliability of the data. If this trend turns out to be the case, though, this may indicate that PTGS2 is involved in the epithelial to mesenchymal transition to some extent. In future work, we aim to test anchorage-independent growth by performing a soft agar colony formation assay and determine if the cell cycle is altered to a differing degree with radiation exposure by using flow cytometry.